first of all, I should say that I've been doing this kind of research in myeloma for probably 25 years or so. And when I first got involved in it, and I would present to audiences that didn't know much about myeloma, I would say that myeloma is a disease of the elderly. Recently, I looked in the mirror, and it is a disease of the upper middle age, okay? The other thing is, how many people in the audience have been diagnosed just within the last year? Okay, and then let's go the other extreme. Anybody diagnosed over 10 years ago? Oh, look at that. That's terrific. That's terrific. So I'm going to talk about a perspective that I have as a researcher, and one of the things that you're going to learn from this talk is I'm going to do science, so we're in science class, and in doing the science, we're going to define myeloma, but for many people, you kind of define myeloma like the 6 blind men defining the elephant. We all look at it in our own perspective, and so I will give you the disclaimer that my perspective may be different than other speakers who have interest in the immunology of cancer or some of the other components of cancer. My interest is in the genetics of cancer and what we can do with that kind of information in terms of understanding better how myeloma develops and better how we might treat it. So it's generally viewed as a difficult disease to cure. Some patients respond to therapy, some don't. Many patients that do respond to therapy relapse. The relapses are often drug resistant, so a patient that responded to a drug no longer responds to that drug. And the reason has a lot to do with the fact that there's individuality to everybody in this audience. If you look around, you all look different. You look different on the outside, and I'll guarantee you you're a lot different on the inside than the person sitting next to you. Your myeloma is different than the person who has myeloma that may be sitting next to you or 3 seats away from you. Everybody has unique characteristics, primarily because of the genetics. So why is it hard to treat? Well, part of the reason it's hard to treat is just because I said nobody's disease is exactly like everybody else's disease. So everybody's got unique characteristics to their particular myeloma, and it may respond, and somebody else's myeloma doesn't respond. They're diagnosed as the same malignancy, a plasma cell malignancy, but how a cell becomes malignant and becomes a tumor can be very different and different pathways. It's kind of like saying that I can drive from here to Salt Lake City, and there's a lot of different...
I can do this too, so you have 2 mics.
Oh, okay. So if I'm driving to Salt Lake City, there are a lot of routes to get there that will get me to Salt Lake City. So it's the same with malignancies. Cells proliferate, they don't die, and they go places you don't want them to go. Lots of ways cells have of doing that, and we'll talk about some of those. The other problem is that myeloma does evolve. There is evolution in myeloma. So once a person is diagnosed with myeloma, over time, changes occur. And those changes occur which impact how the disease progresses and how it responds to therapy. That makes it somewhat difficult to treat as well. The other thing we're getting a better appreciation for is that once we analyze an individual myeloma population, we now understand that there may be small subpopulations of cells. So it gets even more complicated because not only is your myeloma different than the person on the other side of the room, but within your myeloma, there may be cells that have evolved differently than the rest of the myeloma, which means there's heterogeneity in how an individual myeloma might respond. And relapse is a problem. If we could solve the relapse problem, we'd go a long way in curing this disease. So up to this point, I've said, well, given that, is it possible to understand the unique characteristics of every myeloma and treat the myeloma individually, this idea of precision individualized medicine, and treat it individually as opposed to a group? And in many cases, people are given the standard of therapy, which may be perfectly fine, but the downside of that is everybody gets the same therapy, and then we deal with the ones that don't respond in some other way. It would be nice if we could do a better job of predicting the best therapy up front at the time of diagnosis. And the question then that I've been harping on is then what makes the myeloma, what makes every individual unique? And by now, you should know the answer. It's genetics. So it is the genetic characteristics of you and the myeloma cells that are different. So let's do a little biology here. DNA is the central component of codes everything that's going on in the cell. Boy, that's a big dot, isn't it? Okay, DNA codes for everything that's going on in the cell. It's in the nucleus. It's the sequence of that DNA constitutes all of your genes that tells the cell what it should be doing. That DNA is transcribed into another molecule called RNA. The RNA goes out into the cell where it's translated into a functional entity, like a protein. I make the protein look like a factory, okay? So the factory is telling the cell what it's supposed to be doing. Now, in some cases, the DNA can end up with a change in it, a mutation, so that the mutant protein that's derived from that shuts down. It doesn't perform the right job as the factory. Or possibly, you get too much of the protein. So there's too much of the protein. The protein may be absolutely normal, but that cell's making too much of it. Now imagine if you're making too much of a protein that tells a cell to proliferate and divide. You don't want that. So that's a target to start looking at. The other possibility is the amount of RNA that's being produced off of the DNA goes bonkers, and you end up with a lot of RNA being produced, and that generates a lot of that factory protein. So there are cases in all of these where in myeloma, all of these kind of events can happen. These are events that happen in the myeloma cell that change the sequence of the DNA or change how that DNA is regulated so that it's now producing factories that you don't want. But if we understand the factories, we have a better idea of where to target the therapy. So what have we learned in the past 20 years in sequencing DNA? So everybody's genome now has the capability of having a sequence done, and those bases, those A, G, C, T, those unit bases of DNA, every individual has 3 billion of those, okay? That's a lot of sequence. And it turns out that you and the person sitting next to you has exactly the same sequence of those bases 99.9% of the time. That's a lot of similarity. Heck, if you're comparing your DNA to a chimpanzee or an ape, you're about 97 to 98% identical of sequence to an ape, okay? But think about that 0.1% out of 3 billion. That's 1 million differences between you and the person sitting next to you. So there I'm already giving you an information that says one of the reasons you're unique is because although you're 99.9% human genome, about 1 million bases are different between you and the person probably sitting next to you, unless you have a twin sitting next to you. That means that that genotype results in a phenotype, a trait, and everybody's individually unique. And at the same time, every myeloma cell that gets transformed into a malignancy has certain genetic characteristics that make it unique. And our job is to start identifying those characteristics as biomarkers to attack with therapies. So how do we do this, and what are kind of the technologies that we use in all of this? Well, we can now consider, with all of that kind of information that we now have the capability of getting, that DNA sequence from individuals, how many genetic events need to happen before a cell transforms into myeloma? Is it just one bad gene that throws the whole thing off? Or are there many genes? Are there some common genes? Are there genes that are now not behaving correctly and shared among individuals? Or are there unique genes that might be unique to one individual that another individual doesn't have? Do some of those genetic events affect progression of the disease? Do they affect how a myeloma responds? So I'm now saying, do these genetic variations affect any of these things? And the answer is all of the above. All of the above. So at the level of DNA, we can sequence the DNA of an individual. We can sequence the DNA of a myeloma cell. We can sequence the DNA of lots of different people's myeloma cells and start looking where are the common targets to identify potential therapeutic approaches. So what have we learned in the last 5 years or so in doing this kind of stuff in research situations. There are lots of changes in DNA sequences. Myeloma is considered genetically unstable. What that means is the genetics, the genes, the DNA sequence kind of changes over time. The DNA is relatively unstable. Think about it, every time a cell divides, it has to copy the 3 billion bases of the DNA and pass it on to the next cell. Fortunately, over evolutionary time, DNA replication and passing that sequence on accurately is really good. It's done it really well over time. But it's not perfect. It's not perfect. It makes mistakes once in a while. And in myeloma, those mistakes seem to start piling up. Some DNA changes are common. There are certain genes that I'll comment on that seem to be abnormal in many myelomas, but not all myelomas. Some of these changes are associated with high risk. So already now I'm saying that we actually know there are certain genes, genetic abnormalities in a myeloma cell that are associated with high-risk myeloma. So I've now defined a potential biomarker of risk for progression of the disease. And some of the genetic changes that occur in myeloma occur at relapse. You don't see them at the diagnosis, but now things have changed, and now the relapse myeloma has changed some of its genetics. And again, it's studying that kind of transition, the evolution of myeloma, that gives us better ideas about how to start treating it. So the take-home lesson is myeloma is a complicated genetic disease. I gave you lots of information about how the genetics is being altered in these cells in many different ways. I'm going to show you the next slide that would probably have you screaming and running out of the room because it's a scary slide. The next slide is a slide that I asked a graduate student in my lab as we were putting together a review article about treatment of myeloma and ways in which myeloma becomes resistant to therapy. And so I said to the graduate student, I said, I want you to put together a picture. And in that picture, I want you to represent the genes, the paths, the molecules that are all been studied in myeloma as being deregulated or abnormally regulated. I want you to link that to some of the therapies and some of the ways that it's resistant. Okay? So bear with me. That's what he ended up with. Okay, so there's good news, bad news on this. It confirms the fact that myeloma is kind of complicated. Every one of those lines, those arrows, those molecules have been implicated in some way in myeloma, either in its initiating or in its progression or in its therapeutic response. So what's the good news? The good news is every one of these has been studied. There are researchers, the grad student didn't make this up. He pulled this from the literature of people studying all of this. And as a result of that, even though myeloma has a lot of complexity, we're in the phase now of better understanding how all of these molecules are impacting the myeloma growth and potential response to therapy. And some of these may be more important for certain therapies than others. And part of the research now is putting all of this information together. So one of the things that is a truth that I will tell you, another take-home lesson, the databases are getting very, very large. And the thing is that the person that I value the most in our research enterprise, and the people that have marketing skills that can pretty much just get a job anywhere they want, are the people who have computational skills, that they can run computer programs to start making sense of all of that kind of data. And fortunately, we've had some people in the lab who have been really good at that, and I'll show you some examples. So this basically says your medical problems are more complicated than I thought. I'm going to refer you to another doctor who has more medical insurance than I do. Okay? So let's go back to this myeloma genetics. So we start with that cell, and let's take kind of the 30,000-foot view of DNA. And the 30,000-foot view of DNA is your chromosomes. So DNA is organized into these units that we call chromosomes. And you can see that in the normal human chromosome content, every cell has 23 chromosomes, or 24 if it's X and Y. So you have the X and Y, which are sex determining. And then there are 22 other chromosomes, and you'll notice there's a pair. And that's because you get one chromosome from mom and one chromosome from dad. Okay, so you are indeed a hybrid of mom and dad. You can also pretty much blame a fair amount of your genetics on your mother and your father. Okay, and you know, I said some of it, not all of it, because there are things you can do to change your genetics. Things you eat, the sunlight you expose yourself to, the environmental toxins you may not even know about, some of those actually start changing DNA sequence. Okay, so you can't blame it all on mom and dad. The other problem I said was that when DNA is being replicated, think about it, a sperm and an egg, two cells join together, and from that generate a human which now has over 10 trillion cells. That means you had to replicate that sequence 10 trillion times to get that many cells. And as I said, DNA replication and copying that sequence is really good, but it's not perfect. And I dare say if I asked you to write down 3 billion base pairs a bunch of times, you're going to make a mistake once in a while, even if you do have an eraser. Okay? So it does make mistakes. And so the other part of genetics, which is really the hard part to really come to grips with, is when you ask the question, mom and dad, neither of them had myeloma. Why do I have myeloma? And part of the answer is it could be the combination of those two chromosomes now as a hybrid that puts you at higher risk. The other possibility is environmental exposures that you've had you don't even know about that could have interfered with the DNA sequencing and replication. And then the third one, which is really a tough one, it's kind of just bad luck. That is, every once in a while, replication of DNA makes mistakes. It's not perfect. And most of the time, those mistakes you'll never know about. But every once in a while, a mistake is put in a gene that you'd rather not have a mistake in, a gene that maybe now activates proliferation. So that's part of it. So there's a normal chromosome content. So from the chromosome, we can look for, are there any abnormalities just at that level? Now we can also look at the DNA sequence. We can look at the amount of DNA that's making RNA, and we can look at the protein. So these are the genomics, the proteomics, the metabolomics. There are lots of omics studies that are looking at the patterns of the expression of DNA, RNA, protein, for example. And we can ask, are there chromosomal abnormalities that might be found in myeloma? Are there mutations in individual genes that might be found in myeloma? And are there changes in how those genes are expressed, how much RNA is produced out of that, that might be unique to myeloma? And how do those associate with disease progression and response? So here's a chromosomal picture, and you can see on the left is that normal chromosome karyotype. So we have cytogeneticists whose job is to look at chromosomes. And that's what they do in the medical facilities. And I dare say every one of you with myeloma has had a chromosomal content done. And you can see the one on the right is not a normal chromosome content. It's got extra chromosome 1, extra chromosome 3. So one of the things that can happen in myeloma is that you end up with too many chromosomes in a cell. And when you end up with too many, you can have a deregulated cell that can become malignant. The other possibility are chromosomal translocations. And again, many of you have heard you may have a 414 or an 1114 translocation. What that means is in plasma cells, there is a normal function. The normal function is to make antibodies. That's what plasma cells do. It's part of their immune function. They make antibodies. And one of the curious features of antibody genes is in order to express an antibody gene, part of the normal function of a normal cell that makes antibodies is that it has to break the DNA and rejoin gene segments together to make a unique copy of genes. There are not a lot of genes that require that, but antibody genes do. But I introduced a concept here. The DNA has to get broken. So the DNA breaks and genes rearrange, form an antibody gene, and you're good to go. It's a normal function. But breaking DNA, it kind of makes you susceptible to this kind of thing, where you have two chromosomes that break, and then they rejoin, but they don't rejoin back to the original. They rejoin across each other. And so what now I have here is chromosome A and B are now translocated, so a segment of one chromosome joins to another chromosome. That's not normal. And again, in many cases, it doesn't really have much of an impact. But imagine if you have a gene that's really active in chromosome A, and it gets put next to a gene that's not very active, but it causes that gene to get turned on because of its proximity. And so that can happen in these chromosomal translocations where genes get deregulated because new pieces of chromosome are being put next to each other. And if you look at sort of data on this, this is called a circus plot, not to be confused with the clown circus, but this is, and I didn't spell it wrong, that's how it's spelled. It's called a circus plot. And basically what it's showing is that in myeloma, every one of those rainbow lines shows two genes that end up getting put next to each other as a result of a chromosomal break. So you can see IGH on chromosome 14 can break and join with CCND1 on chromosome 11. IGH is a very active gene, and when it's put in proximity of the CCND1, it activates that too. And CCND1, it's a proliferation gene. Okay? So these are examples of the things that are seen in myeloma. Not every myeloma has every one of these. And that's already now, what is the distinction between a myeloma that might have an 11-14 versus a myeloma that might have a 4-14 translocation? So we can now start subgrouping individual myeloma patient samples based on their chromosomal abnormalities that they may have. And there's been a lot of research on that. This is a very busy slide, and you'll only be tested on the middle part of it. So this basically is showing the abnormality, and you can see there's a T414, there's a T1416, and over the years we've learned that certain of these chromosomal abnormalities present increased risk for progression, moderate risk for progression, or slight risk for progression, and they're associated with that. And I'm not going to spend a lot of time in this because Dr. Davies, in one of the next talks, is going to talk about how these kind of chromosomal abnormalities can not only define a distinct characteristic of a myeloma, but it actually might be useful in directing certain therapies. Likewise, individual genes. So we look at individual genes. And one of the genes that's mutated in many, but not all myelomas is a gene called RAS. Now, I'm not going to go through the whole function of RAS, but one of the things RAS does when it's active, it signals through the cell and turns on genes that cause proliferation. Okay? And if you have a mutant form of RAS, there are certain mutant forms where the DNA sequence is different, and the protein that's produced is always on. It never shuts off. And you'll notice that if you look at the KRAS and the NRAS and combine those, about 40 to 50% of myelomas have those RAS mutations. What's the significance of that? And in fact, it's been a little bit difficult to sort all of that out, but we do know that it can impact therapy. But just as important, there's a lot of activity to try to develop therapeutics that target those genes. Okay? And I know Amgen has one that now targets KRAS that we're real interested in. This is another one of those diagrams you're going to go screaming from the room. But it's a diagram that the bottom line I just want to tell you is that if you look from left to right, that's sort of myeloma progression from a mucus monoclonal gammopathy to a smoldering to an active myeloma to a very progressive myeloma. And the one thing you should take home from this slide is that notice that the number of genetic events that are occurring during progression are getting bigger and bigger and bigger. This is part of that evolution. Part of as the myeloma goes along over time, you might have therapeutic response, but the genetic instability starts creating more and more of these kind of genetic events. This is the kind of thing we do in the laboratory. What we do in the laboratory is we do the kind of things where we're looking at the differential expression of genes. So we want to know, how do genes get expressed in a myeloma versus a normal plasma cell? And this is work from John Shaughnessy when he was at Arkansas, and this is called a heat map. And basically, as complicated as that look, it really is somewhat simple, and that is every column down is a sample from a patient. Every row is a different gene. And red means the gene is up, being highly expressed. Green means the gene is being suppressed and not expressed. okay? And you'll notice that on the left in the green bar, those are all normal plasma cells. And on the right are malignant plasma cells or myelomas. Big difference in the pattern, right? Big difference in the pattern. So that right off the top says there's really some really common differences in the expression patterns. But take any two columns in the patient samples and they're not exactly the same. There's some that have green where others have red. So even though the pattern, and I always say, you know, if I take my glasses off, that all blurs as sort of a common pattern. But if I put my glasses on and look a little closer, every one of these is a little different. So the genomics now demonstrates how complicated this can be. We can have chromosomal damages, gene mutations, we can have alterations in gene expression. How can we use this information? So this is one, it's complicated, but myeloma is still myeloma. And so they say your cholesterol stay the same, but now we have more data, okay? So these are 120 genes that differ between myeloma and normal plasma cells. One of the things that this could remind you of is a distinction between normal and abnormal. Instead of looking at red and green and genes and all of that, why not consider that a barcode? A grocery store barcode, okay? What the heck, a grocery store barcode. So look at these barcodes. If you look at the bottom of the barcodes, they look very similar. The thickness of the lines, the pattern of the lines look very similar. But when you get up to about the 3 and the 4 up there, there's a little bit of a pattern difference. Okay? So what if we could generate patterns of those gene expressions and associate those with therapeutic response? Biomarkers, predictors of whether you're going to respond or not. Turns out they were very similar. Those two barcodes are both loaves of bread, but one's wheat bread and one's white bread. Okay? One's a myeloma, the other's a myeloma, but this is a myeloma that responds, this is a myeloma that doesn't. Can we do that? John Shaughnessy developed this kind of marker system where he was looking at genes that were being expressed, and he found out of about 17 to 20,000 genes that everybody has, there were about 70 that did a pretty good job of predicting highly progressive disease or low progressive disease. And they're pretty well separated. Now, the good news on that is that he's developing a genetic test that tells people or tells physicians this patient likely has a progressive, highly progressive, high-risk disease versus a low-risk disease. What it doesn't do is it doesn't say what you do about it, okay? So let's go back, and this is stuff we've done in our lab, and that is we've taken a myeloma cell line. So we take it out of the patient, put it in the laboratory, and we treat it with a drug, in this case Velcade. And the diagram there is increasing concentrations of the drug, and you can see that the myeloma cells that we have in the culture being treated with increasing concentrations of the drug is dying. This is a good survival curve. You're killing myeloma cells, okay? So the myeloma is dying. So the question is, if I take 2 myeloma cells from 2 different patients, if I take 5, are they all going to look like that? Absolutely not. So these are 50 different myeloma cells, and you can see increasing drug concentration. Some of them respond really well, and some of them don't respond well at all. So the question is, what's different? What can I look at? I can look at the gene expression patterns and say, can I find a gene expression pattern that is common to the responders and is different than the non-responders? And there's the pattern. So this is another one of those heat maps. In this case, the gene expression was done, and the computer and the postdoc who knew how to program the computer went through all the genes and said, I can find 42 genes that best distinguish responders, the 5 right columns, from the non-responders, the 5 left columns. Left and right, okay? And you can see that the genes that are up and the genes that are down are very different. This has the potential then of saying, if I can read that and a new patient walks in the door and I analyze those 42 genes, and it looks like that, whoops. If it looks like that, well, that pattern looks like the pattern of the responders. That's a good drug to use. Okay, so the idea is to start using this genetic information, associate it with patients that respond or don't respond, identify the distinction, and then use that distinction to determine whether this therapy is the right therapy for an individual patient. And it does work pretty well. What if we could take those 42 genes and instead of analyzing the tumor, we're going to analyze one cell at a time? Because now I'm going to ask the different question. Is every cell in that tumor going to show the same pattern of response as every other cell? And based on what I told you earlier, the answer is probably not. So now we're going to say, can we develop a method in which we can analyze single cells? And there is a technology. There are a number of these. The one we used was, at the time, was Fluidigm. It allows you, in this device, up there, you see that little green dot? That's one cell. And all the channels there are, you can introduce different liquids so that you can lyse the cell, pull out the RNA, isolate that RNA, and look at the gene expression pattern of one cell. And so what the postdoc did is he said, what expression pattern do I want to look at? I want to look at the responder-non-responder 42 gene pattern. And now I'm going to score it. So he develops a score that says, based on my scoring of these genes, 42 genes, I can give you a probability of resistance score. Okay, and he did it from 0 to 1. And this is what it kind of looks like. Just look at the bottom. Those are all the scores for one cell at a time. And it turns out, and I won't go through it, but where the dotted line is across tends to be a resistance in which the cells are unlikely to respond to proteasome inhibitors like Velcade. And in fact, the 3 patients on the left did not respond well to proteasome inhibitors. The 3 patients in the middle did respond well to proteasome inhibitors. What about the 2 on the right? Mixed population. So the thing we now need to do is to ask the question, are these the people that we need to consider might have a subpopulation of cells that don't respond, and they may now cause a relapse? Are these the patients for which a proteasome inhibitor may work for some, but not all cells? Are these people for which the proteasome inhibitor will work for some, but now it makes sense to add a second drug to nail the ones that the proteasome inhibitor didn't get? So those are the kind of things that experimentally a number of the laboratories are doing. So myeloma genetics is complicated. We know there's variation among patients. We know that variation in your genome and your myeloma genome has an influence on the disease progression. We know, I've shown you examples where some gene expression patterns can actually provide biomarkers or predictors of a response. So the idea is that genetics more and more is giving us more information based on that complicated slide, identifying which of that information is most predictive of a therapeutic response. And what I'm hoping is that at some time in the near future, that the clinical trials are going to be designed not drug A versus drug B, let's look back and see how everybody did. What I'd rather say is when a person walks in the door, we do some testing, we look at the biomarkers and make a choice and say, you get drug A, you get drug B. And let's see how that works as a clinical trial. So that's something that we're currently working on. I think Dr. Davies is going to talk more about this, so I'm not going to go into that. So treatment decisions involve a conversation. Researchers certainly listen to your doctor. And as this doctor says, wow, that was very loud, Mr. Trainer, but I said I wanted to hear your heart. Okay? So that's where I'm going to end. Myeloma Science 101 is over. Thank you.